Today, me and Andrew, who's also on Zoom, are going to talk a little bit about like how we've been thinking about these problems at the IPD and some of the progress that we've been making, and also tell you a little bit about like this package that we've put together, and then how you can potentially get involved at the end. That it's still work in progress, but we'll talk a little bit more about that at the end. Like, first of all, just like a high-level intro, and I'll skip over some of this stuff because I think we're very much aligned with a lot of what Simon said. Is there's this sort of design, build, test, and learn loop of protein design. And it's oftentimes done in this way that requires both quite a lot of in silico exploration and then also the wet lab validation. And kind of these exist in this loop where we're constantly learning about how these tools work and also constantly developing the tools in silico as well. But the problem that we're facing today feels very there's a lot of issues today that exist only within in silico, where purely the part of orchestrating all of these tools, putting them together, and then evaluating them and testing them and all of these things feels incredibly laborious. And there's often many problems associated with this process, which can be streamlined quite a bit. And so when we think about what design should look like, you could imagine it would be something like we have these high-level constraints and ideas. We know how to use these intermediate tools, and we do something as simple as just executing each step in tandem according to some predefined config, and then out comes these proteins we're fitting these complex constraints that we're satisfied enough to want to order into the wet lab. But at the moment, it certainly exists in a slightly different state where after each step, you have to do quite a lot of manual work to either filter down the structures or manage the containers and do all of these things, which we'll talk a little bit about now. But the point being is that there's this labor that has to happen after each step because we lack a lot of standardization that allows pipelining to be very easy. And there's a number of root causes of this, which we've been thinking about. And like one of them is the fact that a lot of these models, like we build wrappers around these models, and a lot of these tools end up somewhat opaque. And this is not just true for the ML models, but it's also true for things like PyRosetta and all of these extra sort of scripts that we want to run on top of these standard design protocols. And then there's also just the fact that there's quite a lot of different constraints we're trying to fit when it comes to both building the models, but also building the pipelines, in the sense that designing for proteins is a very complicated task. There's a lot of different ways you can do it. There's a lot of different constraints that you'll want, which we'll also briefly touch on a little bit in a bit. And then, as Simon also talked about, there's an inherent need to store the path through which these proteins have been generated. So things like RMSD and these things require you to know about the source of the protein and where the fold has come from. And something like this is also incredibly tedious to have to code up yourself. And oftentimes when you use tools like Protein MPNN or these AlphaFold, it doesn't give you that explicit path dependency back straight away. And then there's also a problem of scaling, which we hit oftentimes especially when going for very hard targets, where we end up having to do like millions and millions of files, and sometimes it goes up to terabytes. It entirely depends on how difficult the task is, and also how much you want to push the models. But certainly there's this very fundamental scaling problem where we hit a lot of these hard constraints here. And so you oftentimes have to do very effective management of compute resources. And so one of the things that we've done in line with a lot of other people is open source some of these models and try to put them into one repo. So we've tried RFD3, MPNN, and RF3, and we've put this out there as a unified repo for a lot of the design tools that you could need. And you can go use this now, and we've set out some of these design notebooks where it can walk you through step by step how to chain these together. And it's built on this AtomWorks backend, so it allows you to interact very easily with these things through notebooks, and you can view the designs as you go along. And so there's a few pros that this has come with, that it's in a single repo. It's got these pre-built examples, and it works very well for tasks where we know that we can get like reliable results within a certain amount of compute. But there's obviously problems, which Simon also mentioned, which is it may not fit your solution for whether you have a priority-based cluster from your university. You might have a pool of GPUs that you have constant access to, which you want to be using 24/7, or you may want to use cloud compute as well. And then it's also these notebooks become very hard to maintain. There's a lot of bespoke things that can go wrong when people use them, and in general, sharing them can be a difficult thing. And certainly there's something that I also want to touch on, which is it's really difficult to swap out models in these frameworks often. And so this is something that we think about with this, is how can we make something that's useful as the models evolve, that doesn't build too bespokely around any given model. So you could easily swap out something, some new folding oracle or some new design oracle. And then there's also this general case that I'll make is that there's actual things that are not going to be solved by just making better models, which is, for example, we may always want to use MD tools or use Rosetta, these physics-based methods or docking tools, which are independent of the ML domain. And also the fact that there are certain constraints that are just incredibly difficult to model with these deep learning tools. Things like bond angles are very hard to condition the model on. And so likely the best strategy if you want to get a certain bond angle out for a long time will be to generate a lot of structures and then filter those down. And so that means that there's a problem of how do we orchestrate that effectively to make that process much easier for people. And so we're caught between this issue or this difficulty between unscalable, rigid, flexible notebooks here on the right, where we can build very bespoke pipelines, but it's very hard to scale them. And then in contrast with very inflexible sort of monolithic pipelines that pre-compile a lot of the resources and the ways of doing things into the repo. And so, yeah, we're trying to fit a lot of constraints with this. And all of these different design tasks have very different sort of constraints and different pipelines that they require. And so with this kind of work, we're trying to take all of these different examples and put them into one kind of orchestration repo to make things to orchestrate and solve all of these problems in the back end, allowing you to get access to designing all of these different types of proteins. And yeah, so this is the sort of the final motivating slide. And I think there's a lot of different ways that this could go. We want to really push for something that really enables us to do easy design campaigns in parallel and benchmarking in parallel, swapping out these tools and giving us a lot more information about how these tools are performing. And then there's all sorts of HPC and scaling issues, which we also targeting. And then it should also be much easier to share your notebooks or share your setups with other people and reproduce simple design campaigns that have already worked. That's the third motivating factor here. And so with that, I'll let Andrew take over and tell you a little bit about the implementation that we've got.
Awesome. Thanks so much, Jasper. So yeah, I think as we go into this, you kind of see a couple of our guiding high-level principles were accessibility, interoperability, extensibility, reproducibility, and simplicity. And some of these might seem at odds with one another, but hopefully we can convince you we've done all of them. So just at a high level, what we were going for was a simple Python interface where you could compose together steps just the same way you would compose like functions. And we were aiming for this because with this, you can build stuff on top of it. You can build increasingly beautiful user-facing things, but this captures all of the core backend use cases that you would care about. So yeah, if you can go to the next slide, they're going to get into the nitty-gritty a little bit because I think it's worthwhile. So I'm going to walk you through how we've been thinking about what a design pipeline is and should be, and some of our decisions. So yeah, first we can hop on to thinking about the logic and what we're actually trying to do. So we're basically doing these constraint solving problems, but in practice, what they end up being is we have computations and we have computations on certain artifacts. And so we've been calling these artifacts and operations. And the key thing, I think, for protein design is that artifacts are not just protein structures. There are many other types of things that we care about doing computation on, and it's important that we don't couple too strongly to just protein structures. And then operations are just all of the tools that we use to create or modify these artifacts. And basically what we're doing is we're building a computational graph. We have a bunch of inputs that have some constraints. We do some computation on them, and then we create some outputs. And so you get this, if you're interested, there's this canonical bipartite provenance graph that is basically, this is how you can track what computations did, what they consumed, what they produced, and the relationships between artifacts. And so here, what I'm showing you is simple, but it's basically just the micro-scale computational graph, all the individual artifacts and computations that occurred. And yeah, and here you can see we're branching out. From this, we can extract and record the data flow, the relationships between artifacts, and then also the execution flow, which is the relationships between the computations. And once we've captured all of this information, we can have this complete history of the entire process and fully understand its state. So from here, if you can go to the next slide. Yeah, so now we have this simple mental model of this computational graph that we're doing to try and build some protein to satisfy some constraints. And what does the actual interface look like for this? How does the user construct this graph? And so our goal was to simply allow the designer to express their intent in Python by composing functions. specifying what to run, what inputs to use, and what resources to use, all in the same place, all with clean separation of concerns so that each component owns only one thing and that you get this nice composable portable system. What I was showing you before is the sort of micro graph of this computational graph. But when we think about pipelines, we are typically trying to program these pipelines at what I think of as like the macro scale. And so we're typically looking at batched versions of these computations and artifacts. And I think about micro as being about the causality of what's happened, and macro as being about how you control the flow of what you want to happen. And this is how the user typically interfaces with this graph. And this is these pipeline diagrams that we draw, is we draw these macro scale graphs. But we also need to be able to bridge these two things, and we need to be able to bridge them well. And so in order to do that, an effective pipeline system must allow the user full control over both data and execution flow. So that is not just linearly chaining things. Oftentimes we want to benchmark things. So in a single run, it would be nice to be able to run different sequence design or structure design models in parallel. Or we might want to, like Jasper referred to earlier, do something like calculate RMSD, where this is really a non-linear data flow where this motif from earlier in the design pipeline is coming back later in the design pipeline so that you can do an alignment and calculate the RMSD. And so this is a sort of a non-trivial thing when you don't cleanly abstract the provenance graph. Another example of this, and some in the comments have already hinted at this, is oftentimes we want to do things iteratively and dynamically. And we might want to enable recycling, where you have some for loop where you're running between sequence design and structure prediction and iterating over and over again. So these are all examples of what our sort of interface must be able to cleanly capture and program. And so in order to do this, we need some backend engine that can orchestrate tasks and execute tasks. For me, execution is really about making the operations themselves composable. And so here that means taking, this is very much the standards bit that we've been discussing, making the things that are passed between tools understandable by different tools. And so here we made some choices about having basically the framework has some representation of a structure like a pose, but each tool requires different versions of what that looks like. So each tool has to specify how to go from this standardized implementation to its own bespoke format. Then it has to run the computation, and then it has to convert that bespoke output format into the framework's format. But what we get from this, if we do this, is we make the operations composable, but we also get some other things for free. And what's nice about this is actually this is what people are doing right now. If you're doing protein design, you are doing all of these things already just in a bespoke way. And so I think if we set it up right, all you're doing is you're converting that bespoke effort where everyone's writing their own pre and post processing into someone does it once and then it works. But what you also get from this is some additional background from the framework. So if you can go to the next slide. If we have standardized execution, we also can get standardized provenance for free. We can figure out what's related to what produced what. And we can, yeah, and that's critically important for orchestration. So now that we have the building blocks for what makes these things composable, now we need ways of actually composing them. And that's the job of an orchestrator. We evaluated a bunch of different tools for this and found a lot of the common tools very inadequate. SnakeMake and Nextflow both just don't really, they're too rigid, or they don't meet some of these other complicated constraints that Jasper mentioned earlier, particularly around the size and scale of protein design. So what we've landed on is a combination of this really beautiful orchestration package called Prefect, which is totally Pythonic, and some custom backend that manages and centralizes all the data. So we get this complete provenance, but in a very flexible and dynamic way. And yeah, what's cool about this is now your orchestration code just becomes standard Python code, and you can do it in lots of different ways. So here's an example where we just, much like Simon was talking about earlier, you can just put some of these models in a for loop and they run, and they choose what compute backend to run on based on what you've specified. You can dynamically filter and add or remove things. And so now the entire process of protein design becomes algorithmic. The other thing we've implemented here is we've tried really hard to make it so that it's portable across different interfaces. Different users have different use cases where someone might want to write a script and just let it run and come back a week later, and our entire pipeline is predefined. And others might want to step through the pipeline one by one in a notebook and submit a step, wait, come back, analyze the results by hand. And so I think doing this well means doing this well for both of these cases. The last bit that I haven't told you here is that we have to actually capture all the information that's being generated here. And this is basically, we have to capture the artifacts in the graph provenance itself. Leaning on this sort of computational graph abstraction actually makes it really trivial to figure out how do we store this information, because simply we just store the graph, and from that we can reconstruct anything that we need for the pipeline. So if we capture all of the sort of operations, the instances of the operations, all of the artifacts themselves, and then all of the relationships between them, at any point in time you can restart the pipeline or start from an earlier step or reroute data from earlier in the pipeline. And this was made possible by using a data lakehouse. So we've leaned into using Delta Lake, which is basically just a database of a kind, and it helps us store all the information in a very accessible but also efficient way. So you can centralize all of your structures in one place. You don't have millions of files on the file system. You have associations between all of the different types of things that you might have. So metrics for those structures, sequences for those structures can all be stored cleanly, as well as the relationships between them. And what's nice about Delta Lake is it's basically just, you can interact with it basically like you would interact with a data frame, like pandas. We're using a library called polars here, but it makes the database very accessible. The other nice thing is that it allows centralization of all of the data and information from the pipeline. So you can really easily build on top of this. And here's a simple molestar viewer in a Jupyter notebook where you can load all the structures from a given pipeline step and switch between steps and quickly iterate between them. And so this is something that is early in development, but is already proving useful. So with that sort of covers all of the core components here. And I hope to have convinced you that we're at least getting closer on all of our core design goals of like accessibility, extensibility, interoperability, reproducibility, and simplicity. The last thing I wanted to comment on here is just to reiterate opportunity to give our opinion of what a design workflow system should be. I really think it should be agnostic to all of the different tools. I think strongly coupling it to one tool is definitely a mistake. It should be accessible, so it should be useful. It should provide benefit to both beginners and experts, but also designers and model developers. It should be very expressive. It should not be focused on binders or enzymes, but it should be able to do protein design broadly, and it should capture the complex logic of protein design. It should also be traceable. We should be able to, again, see the history of all of the objects that are created. It should be easily extensible. So to reiterate my point from earlier, I think that a protein structure is an insufficient sort of thing to base your protein design workflow around. There are other things that are critical, and the workflow, I think, should be extensible for that reason. It should obviously be composable, and then, yeah, scalable. I think it should handle scale from I just want to do one test protein and be able to work with it easily, all the way up to I ran 1,000,000 diffusion trajectories, and I think it should handle all of those cleanly. So yeah, we spent a lot of effort on this, and we're really hoping to get it out soon. We don't know exactly when that is, but we think within the next month is reasonable. And we're actively beta testing in our own lab. If you're really excited about it and want to beta test, you can reach out and let us know. But otherwise, hopefully can play around with it soon once it's actually out. So with that, thanks so much for your attention. Happy to take questions.
Great. Thank you very much for this very interesting presentation. It's so fun to see basically how everybody is doing the same thing. If you look at all of the motivations from the previous talks, like the first 10 slides are almost identical in all of them. So I think we're onto something here, definitely. The solutions differ, of course. And I think, again, this part that you mentioned about not the orchestration part, but going from inputs to a standard format back to the inputs, that's sort of probably key in all of the frameworks. And we could like get together here and write this once for everybody, maybe. So let's start with some questions. Actually, I'll start with a very concrete question. For example, let's say you're using RF diffusion to scaffold something, and then in the next step, you want to use protein MPNN to redesign just the scaffold part. And then in the next step, you want to run AlphaFold on that, let's say 3, whatever. And then you want to get just the RMSD of the scaffold part. How would you do that? How do you envision doing that with the engines and the steps that you outlined here?
Yeah. So this is one of the key things why I love this abstraction of like different artifacts, not just a structure, because it decouples like what the structure is from other requirements. And so here we can implement, we have obviously our representation of what a structure is, but we can also have a representation of a generic structure annotation that is separate. And that structure annotation can then be used across all of the different tools. So here you can annotate your motif before you put into RFD3 or whatever your tool is. You run diffusion, you generate your structure, it updates where the annotation is, and then it goes into the next tool and you specify how that annotation gets used in that next tool. And so you basically upfront provide this semantic label of here is my motif. This is the thing that is static and is not redesigned. You can then infer, okay, here's what needs to be sequence designed by MPNN. And then again, at the end, after you've gotten through AlphaFold, you want to do your RMSD calculation. You now have this same semantic label on all of the structures that you've created that you can then say, oh, I want to align based on my motif, calculate RMSD, calculate these distances and angles, etc. And yeah, that's how it works in this framework.
So yeah,
Sorry.
go ahead. It has to be supported.
You first, sorry. Yeah.
Sorry, there's a lag. Sorry. This annotation has to be supported by all the steps then. So I think what you described is what a residue selector is in Rosetta scripts, right? In a sense.
Yep.
So I think it's super important to have these objects as well, supported by all the steps. Yeah, go ahead, Jesper.
Yeah, I was just basically going to say that. I think it's a part of the standardization is thinking about what is a motif. And also sometimes like people want to do more complicated stuff, like select interface residues of interest, or they want to do like hydrophobic residues near the ligand or some crazy stuff like that, right? And having some notion of something that's rigid enough, as you say, like a motif or binder or target annotations, but also flexible enough to support something like wildly different, like the hydrophobic residues near a ligand. And then I guess the second thing is also this complicated problem that is like mostly for enzyme design at the moment, which is when we like, I could have added a slide for this, it might have been easier to explain, but when we have active site residues and we scaffold them, they're unindexed. The position for which the motif ends up in the generated protein is different for every protein you generate. And so that's something we've been thinking about a lot, is making sure that we capture that dependency and make sure that, for example, when we then throw it into MPNN and so on, that we appropriately are fixing that residue or interacting or calculating metrics on that residue. So yeah, that's just, it's like a higher order provenance tracking, which we also think about in this framework.
Yeah, but even if you don't do active residues, even if you just make a new scaffold between two domains, you still get the same problem, especially if you in your config you allow the change of length, then you still have this sort of floating indexing problem, which is easy to solve, but just a lot of tedious bookkeeping in a sense. Yeah.
Yeah. I think here what this framework pushes you to do is you do that once. So whoever writes RF diffusion, there are these unindexed sequences that change and it outputs some mapping, and then you update that in this post-processing step. So yeah, I think it does get challenging, but I think the whole goal is to set up something where someone does it once and then it just propagates well to everyone.
Exactly.
There's a couple of questions that I wanted to answer from the chat.
Yeah, go for it.
So,
I wasn't reading the chat.
How dependency management works. So currently what we have is we have this orchestration environment that doesn't run any of the protein design tools itself. It just simply is an environment to run the orchestration. And then all of the tools themselves are run in containers or specific Conda environments or whatever you might have set up for those. And so the dependencies of the tool and the orchestrator are decoupled. There's a question about when will this be released as an open source platform, full demo use cases, antibody binders, still pipelines, the agentic AI. You could imagine agents composing tools and pipelines, which yes, very astute observation. I think if someone were to build a pipeline system that is like well abstracted and set it up with an agent and an MCP server and tools and skills, that yes, you would be able to coax the agent to do this quite well. Maybe a little bit of foreshadowing. The plan is to release this in about a month, but we'll see depending on just how much effort it takes us to get it to a robust state where we're happy with it. It should be pretty soon. We intend to release it with some demo use cases. I don't know exactly what those will be, but hopefully at least a protein binder, a small molecule binder, and maybe an enzyme design use case, something like that would be nice.